Hello student, Bhanu Solanki again in continuity of NEET 2020 genetics topic. In this episode we will continue our terminology and rest of the part of the genetics. So continue. This time we will discuss about the homologous chromosome and non-homologous chromosome. Homologous chromosome, you are watching here there two sets of chromosome, similar size, similar shape, similar appearance, and here there I am writing some genes. The locus, the position of the gene is also same, means same size, same shape and same locus of the genes. Genes may be same as capital D or may be different as capital A small a. So gene is not the parameter, locus, size and shape are the parameters. So those chromosomes that are similar in size, shape and locus of the gene are known as a homologous chromosomes. It means this pair of the chromosome is a homologous pair of chromosome. Similarly here there another pair you are watching, suppose P, R, Q, T. This is the another pair of chromosome that is also similar in size, similar in shape and similar about the locus of the genes. Genes may be same or may not be, same, may not be. So this is the another set of homologous chromosome. So there are two homologous chromosomes you are watching, this homologous chromosome and this is another homologous pair of chromosome. Now you are watching that there are genes are present. Now look at this set of chromosome. They are not same in size, they are not same in shape and their locus of the genes are also different. It means they are considered as non-homologous chromosomes, but they are homologous. This one is also homologous. Here there this is homologous, this is also homologous. So homologous pair of chromosome. Now the most crucial part that I am going to discuss with you, this one and this one is a pair of the genes. Similarly, this one and this one is also a pair of genes. This one and this one is also a pair of gene. And here also you can watch pair of gene. Look at here, gene. The term is a gene, means any segment of the DNA. Functional segment of the DNA is known as a gene. Functional segment in terms of what? Those segment of the DNA that either directly or indirectly regulate to the character are known as a gene. Means it is not necessary that the DNA segment that give rise to a protein product is only considered as a gene. No. If they are giving rise to a protein product, then also they are gene. If they are giving rise only to the RNA but not to the protein, then also they are a gene. And even if they are neither producing RNA and nor they are producing any kind of a protein product, even after that, if they are taking part into the regulation of a character, then they are also considered as a gene. So any of the segment of the DNA, that regulate to the inheritance of a character, expression of a character is collectively known as a gene. So this is gene. Now here there another term that I am going to use is the allele. Allele. The word allele actually arises from the word allelomorph. Synonymously you can use them, allele or allelomorph. you can see that gene A, capital A and small a are regulating to the same character, like height of the plant or shape of the seed or the flower color. Means these two genes, capital A and small a, belong to the same character. Second thing, they belong to the homologous chromosome, same homologous chromosome, yes, and they belong to the same locus also, yes. Means those genes which belong to the same chromosome, belong to the same locus and belong to the same character. Trait may be same or may be different, not an issue. Those genes which share to the same chromosome, same locus and same character are known as alleles. So capital A and small a are allelic genes? Yes, they are allelic genes. Capital B and small b is also allelic genes. But now look, this combination, capital D and small c, they are present on the same homologous chromosome. Yes, this is true, but neither they are sharing to the common locus nor they are regulating to the same character. While in the introductory part I already said that the allele term for the gene we can use only if they are belonging to these three aspects: same chromosome, same locus, same character. Here chromosome is same in case of the D and C combination, but locus and the character are different. So these are not considered as a genes, I mean allelic genes, they are considered as a non-allelic gene. Same is the situation with the combination D and T also. You know, D and T belong to the different chromosomes, different locus, different character, so they are also non-allelic. So this pair is non-allelic, this pair is also non-allelic. So I would like to conclude that allelic genes are always present on homologous chromosome, yes, but all those genes which are present on the homologous chromosome are not allelic. They may be allelic or they may not be. They can be considered as a allelic only if they are following to the three common conditions: common chromosome, common locus, common character. So this is the allelic gene, this is the allelic gene, this is the non-allelic gene, and this is also non-allelic gene. So allelic genes are always present on homologous chromosome. All the genes present on homologous chromosome are not allelic. They may be or may not be. But all those genes which are present on the different chromosome are always non-allelic. Yes. So one more thing I would like to say, that allele and gene term, how we can forget that all the alleles are genes. All the alleles are genes, but all the genes are not alleles. D and C are also two genes, but they are not the two alleles. A and A are also two genes, but they are two alleles also. So this is the allelic gene, means these are the genes, these are the alleles. These are the genes, but these are not the alleles. So all alleles are gene, but all genes are not alleles. You can use the word allele only in condition common chromosome, common locus, common character. That's it. So our discussion comes to the point alleles, allelic genes or allelomorphs. Now allelic genes can show two types of combinations: homozygous and heterozygous. Means you can say allelic genes are of two types. Allelic genes show two types of combinations: homozygous and heterozygous. Homozygous means identical allelic gene pair, means capital A, it is a homozygous condition, or small a is a homozygous condition, but capital A, small a is the heterozygous condition. Capital B is a homozygous condition, small b is a homozygous condition, but capital B, small b is the heterozygous condition. So allelic genes may be homozygous or may be heterozygous. One more terminology I would like to share with the students, that is the hemizygous condition. Hemizygous condition. You see, this is our suppose X chromosome, and suppose this is Y chromosome. Here there is a capital A, here there is a small a, here there is a capital B, here there is a capital B, here there is a capital C, there is a capital C, there is suppose T, there is a P, there is a Q, there is a S. You must notice here there that this part of the X chromosome and this part of the Y chromosome, suppose this is the Y chromosome, this is associated with the common locus and common type of a gene. It means this portion is the homologous portion, but this is non-homologous portion, this is non-homologous portion, and this is also non-homologous portion. So this gene P and Q and T and S, gene P, Q, T and S, these genes, these four genes are present in a diploid organism, where their X chromosome as well as Y chromosome is present. But these four chromosomes, these four genes are such kind of genes which would be present in a single copy. Whenever any gene is present in a single copy in a diploid cell, that condition is known as a hemizygous condition. So all the genes which are present in the non-homologous region of the X chromosome and Y chromosome are simply example of hemizygous condition. I repeat again, whenever any gene present in a single copy within a diploid cell or diploid organism, that is entitled as the hemizygous condition. So allelic genes may be homozygous, may be heterozygous, and sometimes they may show the condition hemizygous also. Next, few more terms we have to discuss. This time pure line or true breed. It means group of the homozygous organisms arise by repeated selfing in self-pollinating plants called pure line or true breed. I repeat, pure line or true breed is a group of homozygous organisms. And these homozygous organisms arise how? They arise by the repeated selfing, repeated self-pollination. In a self-pollinating plant, the plant should carry the tendency of the self-pollination, and in such a self-pollinating plant, if we do repeatedly selfing, then we will obtain a group of the homozygous organism. That group of the homozygous organism is known as a pure line or true breed. A similar kind of a word is there that is called inbreed line. Group of homozygous organisms arise by crossing between closely related members. Means pure line is a group of homozygous organism, inbreed line is also group of the homozygous organism, but it arises by the selfing and it arises by the crossing. It arises by the repeated selfing in a self-pollinating plants. It arises by the crossing in between the two related, very closely related members. But condition is that it should be a cross. It should be a cross pollination, not a self pollination. Then in this condition, if you obtain a group of homozygous organism, then we can't use the word pure line or true breed. Then the technical term is an inbreed line. So inbreed line is also a group of homozygous organism like true breed or pure line, but it arises by the crossing between closely related members. The process of the production of inbreed line is known as inbreeding. Now you can easily conclude that if we are doing inbreeding, then definitely a group of the homozygous organism arise. It means inbreeding always result into the homozygosity. In AMS 2015 there was a question. I would like to share with you. The question was that inbreeding results into. First option was the homozygosity and second option was the inbreeding depression. Inbreeding depression means when the vigor of the species or population decrease, means inferior population whenever arise after any cross between the closely related members, that is called inbreeding depression, means decrease in the vigor. But you should remember, decrease in vigor can occur only in one condition, and the condition is that whenever homozygosity appear for the recessive genes, means after cross if I obtain small a combination, then it is an inbreeding depression. But a homozygosity can also be in terms of the capital A. In this condition, the vigor, the power, the strength will not decrease. So it is also homozygosity. It is also a consequence of inbreeding. It is also a consequence of the inbreeding. But this is not the case of inbreeding depression. This is the case of inbreeding depression. So inbreeding not always result into the inbreeding depression, but inbreeding always result into the homozygosity. So here there in this question of the AIMS, the answer was the homozygosity, not the inbreeding depression. Anyway, so inbreed line and true breed, both of the similar kind of a words but technically different, that you now become familiar about this technicality. Next in the terminology is the forward genetics and reverse genetics. Forward genetics means whenever we move from the phenotype to genotype, it is called forward genetics. Means for a known phenotype, when we determine the genetic constitution of this phenotype, that kind of a study is known as a forward genetics. Mendelian genetics was one of the example of forward genetics. Second is the reverse genetics. Where there, if we move from the genotype to phenotype, this is called reverse genetics. Means any genetic constitution we know, and for this genetic constitution what protein product or one phenotype it is exhibiting, that thing if we find out, then such kind of a genetics is known as a reverse genetics. You have studied about the Human Genome Project, where there it was observed that around 50% genes of our genome are like those where there the protein product of those genes is still unknown for us. It means if whenever we find out the protein product of these genes, what is our direction of the study? Gene to phenotype, means it is a case of reverse genetics. So forward genetics and reverse genetics respectively, phenotype to genotype forward genetics and genotype to phenotype is the reverse genetics. Afterwards, one more important aspect I will discuss in the terminology, that is the dominant gene versus recessive gene. Then we will come to the actual aspect of the genetics. Dominance, dominant versus recessive. This aspect is very important and we have to understand it. Suppose this is one of the chromosome in which this is the DNA. This segment of the DNA, suppose, is a gene. I already said to you that every gene is a DNA, but every DNA is not a gene. So this segment of the DNA, suppose, is a gene. The gene which is present, which was present since the origin, means it is the original gene. It is also known as a wild gene. It is also known as an unmodified gene. And it is also known as a dominant gene. I can conclude likewise, the gene which is present since the origin of a species, since the beginning, that gene is known as the original gene or wild gene or unmodified gene or dominant gene. This gene, suppose I give it as a symbol capital A, it will give rise to a particular enzyme, and this particular enzyme lead to a conversion of a particular substrate into a particular product. Conversion of this substrate into a particular protein product depends on the presence of this enzyme, and this enzyme is a result of this gene A, that is the original gene, that is the wild gene, that is the unmodified gene, that is the dominant gene. Now you all are aware that this gene A can go through the process of mutation. And after mutation, it can give rise to the different forms, because we all are aware that the mutations are of various types. There are three basic terminology regarding to the type of mutations I would like to share with you: same sense mutation, missense mutation, and nonsense mutation. Whenever after the modification or alteration within a gene, if their protein product remains same, means the sense not get changed, that kind of a mutation called same sense mutation. If after change in the gene, in the nitrogen based sequence of the gene, if a bit of change in terms of a one amino acid suppose change into the protein, or maybe possible more than one gene get changed into the protein product, that kind of mutation is known as a missense mutation. After missense mutation, there may be possible that the gene become less efficient, I mean the enzyme become less efficient, or may be possible that the enzyme become non-functional. One more possibility is there, although that it is also possible that the enzyme become more efficient, but you all are aware that most of the time mutations are harmful. So the third possibility, means the enzyme become more efficient, is almost negligible. So with respect to the missense mutation, when within a protein one or two amino acid get changed, it can result into the most of the time either less efficient enzyme or non-functional enzyme. Third type of a mutation is a nonsense mutation, means after mutation, if the sense of DNA finish, then it is called nonsense mutation. In simple terms, I can say after mutation, if any functional code convert into the stop codon or nonsense codon, then such kind of a mutation called nonsense mutation. It means whenever nonsense mutation occur, there would be no protein product, because stop codon means what? Those genetic codes that stop the process of protein synthesis, nonsense codons or the stop codons. Means here there four possibilities I will share with you with respect to the mutation within a wild gene. Same sense mutation, missense mutation, missense mutation, and nonsense mutation. After same sense mutation, same enzyme will arise. After missense mutation, either less efficient enzyme arise or non-functional enzyme arise, or no enzyme arise. These are the four possibilities. Okay, same sense mutation, then the same enzyme will arise. Missense mutation, then the less efficient enzyme will arise. Missense mutation can also result into the non-functional enzyme, or and nonsense mutation will result into the no enzyme production at all. Now, in this condition, you all are aware that the substrate will transform into the product. Yes, because the same enzyme arise. In that condition, although efficiency become less, it means in the unit time, suppose initially X number of products were arising. Now after this mutation, it is producing X minus 1 protein product, but it is still producing the protein product. Now it means in this condition also substrate to product formation will occur, only the efficiency, the rate of the protein production will affected. But in case of missense mutation, substrate cannot to be transformed into the product. And if no enzyme, then also substrate cannot to be converted into the product. It means these two variant of the gene, are similar to the original or wild gene. It means they are also considered as a dominant. But these two genes or the alternative forms, mutated forms of the gene cannot convert the substrate into the product. It means they would be considered as a recessive. So I would like to say that the dominant gene may be original or wild gene, may be unmodified gene, or sometimes it may be modified gene but equivalent to the unmodified gene. So dominant genes may be unmodified or may be those modified which are equivalent to the unmodified, means the case 1 and 2. Means after watching any gene or gene product, you cannot say strictly that it is unmodified. It may be unmodified or it is equivalent to the unmodified. But those which are producing to the recessive phenotype, those kind of genes are considered as a recessive gene because they are not capable to transform substrate into the product. So recessive genes are the modified genes. Dominant genes may be unmodified or equivalent to the modified. So dominant genes may be unmodified or equivalent to the unmodified, but recessive genes are the modified genes. Dominant gene, in case of the dominant gene, they can express in both homozygous condition, capital A, as well as heterozygous condition, means capital A, small a. Homozygous condition as well as in heterozygous condition, in both of the condition they can express. You know why? Because in both of the condition, capital A is present, it means they are capable to transform the substrate into the protein product. But recessive gene will express only in the homozygous condition, means small a, small a. Here there, you should also be careful about this aspect that you know that phenotype, whether it is a dominant phenotype or it is a recessive phenotype, both kind of a phenotype are the consequence of dominant allele or dominant gene. I repeat again, dominant phenotype as well as recessive phenotype, both are the consequence of dominant allele. How I can say likewise? Yes, I explain it. Dominant phenotype is due to the presence of dominant gene, and recessive phenotype is not due to the presence of a recessive gene, it is due to the lack of dominant gene. I can say in simple words likewise, tall plant is tall because it is a tall, but dwarf plant is a dwarf because it is not at all. Means tallness is due to the presence of capital T and dwarfness is due to the absence of capital T. Violet flower color is due to the presence of the dominant gene, and white flower color is not because of the presence of a recessive gene, it is because of the lack of dominant gene. In case of human being Human males are male because they are male. Human females are female because they are not the male. Because for the femaleness there is no gene. Y gene, Y chromosome. On the Y chromosome there is a presence of a testis determining factor gene, TDF gene, that is also called master gene. When master gene present, the fetus will convert into the male. But if master gene is absent, it will convert into the female. So maleness is due to the presence of TDF gene and femaleness is because of the absence of TDF gene. So in our NCERT it is given that phenotype is due to the presence of dominant allele as well as because of the absence of dominant allele. So this is all about the terminology, students. Now you are in a comfortable zone with respect to these kind of words: dominant, recessive, gene, allele, homozygous, heterozygous, homologous, non-homologous. Afterwards, the study of the genetics become very convenient because now you are familiar, you are master about the terminology. So the actual genetics and the aspects of the genetics we will discuss into the coming episode. Thank you very much.